Dr. Leonid Brodsky, as already mentioned, is the director of the Tauber Bioinformatics Research Center at University of Haifa. His background is mathematics and biostatistics, but today he is going to work, he's going to share with us about mathematical modeling of single-stranded RNA viruses, the role of naturally occurring defective virus genomes, as well as other topics as well. So Dr. Brodsky, I will pass this on to you.
Okay, can you hear me?
Yes, we can hear you.
Okay.
And see the screen. Yeah.
Actually, I have two topics. The first one is virology. Actually, we did it together with Professor Andina. And so this is ordinary differential equation modeling of an interplay between some flavivirus with defective genomes, defective viral genomes of this virus, which appear naturally. Okay, so the defective genome, here's the genome of this virus, flavivirus in general. The majority of defective genomes actually eliminate this portion. So structural proteins and NS1, which is very important because it works in trans and participate in replication. This is just a story about modeling, like an example of modeling. So we started with two models. One model is simplified, we call it ODE1, where we consider it just generation of virus particle by cells after infection, and the story when the uninfected cells are infected by virus particles, and so on. So actually the production of virus particle here is constant. It not depends on intracellular story, intracellular processes. Another model, which we call ODE5, takes into consideration this intracellular story. And it generates a lot of difference. Okay, this is just differential equations. You can see that there are two models. They have a shared portion related to cells and individual portions, generation of particles, and intracellular story here, and generation of particles which depends on intracellular story inside cells. So virus replication and both wild type and defective particles, genomes. Okay, what kind of data we have in order to fit model to the data? We had, the experiment was done in two cell cultures, Vero and mosquito cell cultures, differently in 12 passages. And the data which we have at the end to fit the model are virus titer and next generation sequencing data, which also was used here in order to fit the model. Okay, as to ODE5 model, you can see that virus particles, majority is wild type, are perfectly good fit to data here for in raw cell culture and in mosquito cell culture. And as to next generation sequencing data, actually what we did is what is called phylogenomics. It means that we created haplotypes, so to say, consensus of clusters of genomes which appear in, which were assembled from the data, and the request was to fit coalescent based phylogeny with the modeling, with all these processes of creating genomes and eliminating genomes, in order to get good correspondence of coalescent processes, coalescent phylogeny, and tuned to the model. So basically parameters of the model were tuned with this type of data, virus titer, and with this type of data together. The first interesting question is, both models are good for this or not? It appears that the simplified model, ODE1, is not good. Why? First, some important rate, cell birth, cell multiplication of cells parameter was unrealistic. In ODE5, after tuning to the data, the model demonstrated realistic value for this parameter. And here we can see that actually completely unrealistic this model, in order to be fitted to the data, fitted to this type of data, gave a huge amount of uninfected cells as a result. Very different from ODE5 model, which behavior is very good. I mean that just cells are dying. Nothing specific is happening with multiplication of cells. Okay, so obviously it is very important to have this, to have a model of these intracellular processes in order to get reasonable result. Now the question is, what is the difference between this interplay of wild-type viruses and their defective genomes? What's the difference between two cell cultures? With sensitivity type of analysis, we detected key rates, so to say. This is replication rates of both wild type, defective particles, defective genomes, rate of protein production, and rate of production of virus particles. So based on analysis of all 12, actually 11 passages, we found that typically values of these parameters are higher in mosquito. And mosquito is this flavivirus is actually developed mosquito, and mammalian cells are not natural host for them. Second, we try to answer the major question. The major question is, what's the role of defective particles in all of that? What they are doing? Can we say that defective particles are therapeutic? It means that they're saving uninfected cells, or this is not related to any attenuation, so to say, of viral infection. Okay, here what was done is actually a jump from modeling to biology, sort of. We took, based exclusively on this model. First, we performed variations of our parameters of our model. And for each combination of parameters, so parameters are up and down 20%, we got some solution. And this solution looks like that. This is time behavior for here, for instance, is for infected cells. And this solution like that is for genomes, defective genomes in cells infected by defective particles. And this solution is for proteins. So first, based on these observations, of modeling observations, so to say, so the values of these curves, we can create a relationship between solution and the different combination of parameters. And this relationship, based on conditional entropy, could be presented on the SVD plane, like PCA plane. And we can see that these points are solution under different combination of parameters. What is interesting, we have some basic solution. This basic solution when parameters are most fitted to the data, and most deviating in two different directions, most deviating solutions. So the question is, what actually it means, the movement in this direction and in this direction in understandable terms of behavior. So for behavior, we see two components of this behavior: the height of the peak and pace of the process. So this process is faster than this one. So how we can interpret this picture in these terms? And it appears we can. It appears that we can see that all parameters related to defective particles, to processes with defective particles, are in terms of peak height, are lower than parameters associated with wild type. And more than that, parameters which rules behavior of uninfected cells are here in the group of defective parameters, so to say. And when we change the direction, so it will be, there are two directions here, this one and this one. So in this direction, X movement, we have this sort of picture, and in opposite direction, not opposite, but orthogonal direction, we have this sort of picture. What it means in terms of uninfected cells. We can see that uninfected cells are jumping together with parameters related to uninfected cells, are jumping together with parameters associated with defective particles. Like it's depicted here. So we can say, and this is actually what we are saying in the manuscript, that defective particles somehow attenuating behavior of uninfected cells. It means the loss of uninfected cells is attenuated by this defective particle. This is, so to say, a biological conclusion, major biological conclusion from the story, that we found that indeed defective particles somehow helping not to lose too many uninfected cells. Okay, and the here I'm just saying that acknowledgements that this is people from Tauber Center, Professor Andino, and our collaborator from Institut Pasteur in France. Okay, the next story, I have 5 minutes. Yes, Ilya?
Yes, you have some time. Yeah.
Okay. This is another story. This is a story about how to introduce time in bioinformatics analysis of omics data in particular. For instance, single cell gene expression data, or multi-omics and physiological features of individuals, aging, and so on. Metagenomics data. So we have some observation, but can we present it as a time-related potential? So like a ball on the mountain, which can go down. So where we have mountains and where we have sinks. Okay, actually, it was great publication of this sort, and we expanded what was found in this publication, that this potential for observation can be reconstructed using Laplacian matrix. And if we know, for instance, stages like aging stages or single cell differentiation stages for cells. So preparing a graph of some sort in multidimensional space and preparing this Laplacian matrix, we can, for observed cases, we can say what kind of potential, dynamic potential, force field potential we have along these cases. The next stage is, okay, what we can say about the point here inside. And it means that we would like to prepare this surface, which will show that this is a mountain and this is some sink, and this stability state sink could be related, for instance, to old individuals with some illness, and here with just healthy old individuals. Okay, we did it with a technique of partial differential equations, and based on boundary values, we restore this surface. And as a result, with this surface, and this is a projection of this surface, so this is a mountain. Now, for any point, we can say what are possible trajectory and what are times to go and probabilities to go over this trajectory or over that trajectory. And even in the case of going from this hole to this hole over the potential barrier, like here, yes. And we can estimate what time for diffusion is needed in order to go here, from here to here, or in opposite direction. So from data under condition of that the system is stabilized, we can actually restore time story. And since it can be applied to many types of multi-feature data, I believe this is good perspective to introduce this time axis in and actually real biology. It means to the multi-omics study. Okay, this story was done by the team of Tauber Bioinformatics Center in collaboration with Professor Gutkov from Roswell Park Comprehensive Cancer Center, Buffalo. Thank you.
Okay, thank you very much for the presentation. And, you know, I think it follows some of the discussions that we just had. And our next speaker is going to be kind of addressing, I think, some of the biological parts of this story. So, Dr. Andino, I wonder if you had any comments on this presentation that we just saw. Hopefully he's...
Yeah, I was taking notes. It's very interesting. The last topic was, you know, obviously the first topic is something that is very well, we've been discussing this for a long time with Leonid, and I will extend a little bit more that, but it was a great introduction to trying to understand how this defective genomes interfere with the wild type, and you know, can this be an approach for therapy? So when I make my presentation, I'd like to give an update of where we are on that in a manuscript that we are preparing on that and that subject. But I was very interested on the second part. And I have a question, Leonid. So clearly the idea of time resolution of these processes are critical. And I like the idea that you descend in a mountain, from a mountain. And coming down from a mountain, you can take different path, I imagine. And the question is, can we understand that, you know, you're really going down? And can you anticipate what is this path? Maybe we can interrupt this path, and that will be, you know, the more practical exercise here. So how do you train that model? What kind of information can be used to train the model to define better whether there are common paths, if everybody is taking the trail, or people go down the mountains and completely random ways?
Okay, the only thing which you need is some stabilized system and measurement of the system, multi-dimensional measurements. And so the first step is to understand how this dynamic potential behave on observations. And after that, the surface with unknown cases also could be reconstructed. And as to trajectories, we actually can, based on parameters of diffusion, we can estimate probabilities for every trajectory. Based on, of course, based on these parameters. And maybe it will answer your question that some trajectory will be with low probabilities and the other trajectory will be very, very probable.
Mm-hmm.
Of course, in the case of aging is a very probable process, but we have two directions: to be healthy or to be with some illness. And the very important question is to what direction we will go, starting from specific point on the surface. And second, if it's possible, for instance, from illness to go back to health.
Very interesting. Thank you.